Welcome to the Molecules and Minerals podcast. I'm Stephen Goldman. I'm a former trial lawyer, corporate director, executive, and a biotech investor. I'm here today with Eran Ophir, PhD, President and CEO of Compugen Limited. Compugen is a clinical stage therapeutic discovery and development company based in Israel. What differentiates Compugen is its validated AI machine learning computational drug discovery platform called Unigen, which for years now has been using proprietary algorithms to identify novel drug targets and to develop therapeutics in the field of cancer immunotherapies. Compugen currently has 4 clinical stage programs: COM701, a potential first-in-class anti-PVRIG antibody being evaluated by Compugen in a phase 1 and 2 trial as a single agent in a maintenance setting in relapsed platinum-sensitive ovarian cancer, and it's named the MAIA ovarian trial. Secondly, COM902, a potential best-in-class anti-TIGIT antibody evaluated for the treatment of solid tumors in monotherapy and in combination with other immunotherapies. Thirdly, Rilvegostomig, a PD-1 TIGIT bispecific antibody with a TIGIT component derived from Compugen's COM902 program, being developed by AstraZeneca pursuant to an exclusive license between Compugen and AstraZeneca. Fourthly, GS0321, previously named COM503, Compugen's potential first-in-class antibody, which blocks the interaction between IL-18 binding protein and IL-18, and is licensed to Gilead and currently being evaluated in a phase 1 clinical trial in solid tumors, both in monotherapy and in combination with Gilead's PD-1 checkpoint inhibitor. I was first introduced to Compugen in 2017 when I had a long conversation with its chairman and former CEO, Martin Gerstel. Actually, it was more of an incredibly fascinating and wide-ranging lecture by Martin on the science of computational drug discovery. I published my first article about Compugen in February 2018, more than 8 years ago, and bought my first Compugen shares at that time. Compugen began in 1993 as a specialized computer company founded by 3 young scientists. In the mid-1990s, Compugen began an effort to create better algorithms to analyze biological sequence data with a small team of mathematicians, physicists, computer scientists, and biologists, which evolved into a larger multidisciplinary team focused on building mathematical models of the underlying science and establishing a predictive biology capability. In 2009, Compugen started to establish a pipeline of product candidates, initially focusing on discovering novel immune checkpoint regulators. Compugen computationally discovered an immuno-oncology antibody targeting TIGIT in 2009, which it developed into an Fc-reduced anti-TIGIT antibody checkpoint inhibitor called COM902. The discovery was made at or about the same time as TIGIT was discovered by Genentech, now Roche. Compugen's computationally discovered PVRIG, another checkpoint inhibitor called COM701 by Compugen, in 2016. Compugen's business model to date is to selectively enter into collaborations for its novel targets and related drug product candidates at various stages of research and development. Compugen has entered into major partnerships with AstraZeneca and Gilead, where it has the potential to earn $1 billion in various milestone payments in addition to royalties on future commercial sales. Currently, Compugen's 3 most advanced value drivers include, first, its phase 1 and 2 MAIA ovarian cancer trial, and that stands for the maintenance immunotherapy with an anti-PVRIG antibody using an adaptive trial platform, testing Compugen's wholly owned COM701 in 60 patients with relapsed platinum-sensitive ovarian cancer in a maintenance setting. The trial is placebo-controlled on a 2-to-1 ratio of a treatment group to placebo. Compugen is focusing on this maintenance treatment area where no approved treatments exist and has great unmet need. Interim readout of this trial is expected in Q1 of 2027. Depending on the outcome, Compugen will look to the FDA for feedback on a registrational trial and may initiate combination trials with COM701 and other immunotherapies in the future under this trial's adaptive platform. Compugen's second most important value driver is its partnership with AstraZeneca, which is advancing its Fc-reduced TIGIT anti-PD-1 bispecific drug, Rilvegostomig, derived from Compugen's COM902 anti-TIGIT. Rilvegostomig is currently being advanced by AstraZeneca in 11 phase 3 and 14 phase 1 and 2 trials in various solid cancer tumors, including lung, gastrointestinal, endometrial, and other solid tumors. AstraZeneca estimates that Rilvegostomig could generate more than $5 billion in projected peak annual sales, which would in turn generate additional milestone payments and a mid-single-digit royalty for Compugen. Compugen's third most important value driver arises out of its 2023 agreement with Gilead Sciences, under which Compugen has licensed its first-in-class anti-IL-18 antibody protein to Gilead, including Compugen's COM503 drug candidate, now known as GS0321. GS0321 is currently in phase 1 clinical trial development, both in monotherapy and in combination with Gilead's PD-1 checkpoint inhibitor. Gilead has already paid Compugen $90 million, including $60 million upfront and an additional $30 million when the IND cleared to allow this phase 1 trial to proceed. Compugen can earn an additional $758 million from Gilead in development, regulatory, and commercial milestones. In addition, it is entitled to earn high single-digit to low double-digit royalties on worldwide sales of GS0321 once approved. I would now like to introduce Eran Ophir, PhD, who was appointed President and CEO of Compugen Limited in September 2025, after previously being Compugen's Chief Scientific Officer. Eran, thank you for joining me today.
Thanks for having me, and thank you for the great introduction about Compugen. And yeah, about my background, my scientific background is bioinformatics. Initially, actually goes along nicely with the way Compugen are handling combination of biology and data science and computer science. Later on, studied immunology and a PhD in the Weizmann Institute. In Compugen, I joined more than a decade ago. In the last 5 years, I was part of Compugen management. My last position was the CSO, Chief Scientific Officer of Compugen. And as mentioned, since September, I'm the CEO of Compugen.
Terrific. So you've now been at the helm of Compugen for close to 9 months. So what's your vision for the company for the next 1, 3, and 5 years? And has that vision changed since taking over as CEO?
So first, since I was part of the management for the last 5 years, working closely with Anat, the former CEO, on framing the strategy, so my vision for Compugen didn't change. I was part of the team that built the vision, the current vision of Compugen. And in the coming 1 year, I definitely expect to see definitive results showing monotherapy activity of COM701 in the MAIA ovarian study by Q1 27. In this year also, definitely I expect to see evolvement of the anti-IL-18BP program, which is in the clinic now for more than 1 year, evolving of more results supporting Rilvegostomig towards approval along the 11 phase 3 studies. And definitely, while it's not disclosed yet, but we definitely will continue to progress the early pipeline along the coming year. Looking 3 years forward, I think we should be in the position, and again, we're talking about good scenario, in a position that COM701 ideally is in its phase 3 study after showing activity in platinum-sensitive ovarian cancer, maybe in other trials. Rilvegostomig in 3 years from now, hopefully, will already be approved, deriving Compugen for annual milestones and more in royalties. And yeah, of COM503 will be currently GS0321 will be also deep in development, probably already advanced trials if successful. And looking really in the vision in the long term, we are planning to continue and use our validated computational engine, Unigen, and to bring and to establish a pipeline of innovative therapies that could really make a difference to cancer patients.
Terrific. So, which of Compugen's programs today do you see as most valuable, and which do you see as the biggest value drivers for Compugen and its shareholders this year and during the next few years?
Yeah, it's a difficult question because it's like asking which child you like the most. Um, Definitely the COM701 is fully owned assets, and we have a critical readout by Q1 27. On the other hand, we cannot ignore Rilvegostomig, along with 11 phase 3 trials that AstraZeneca are predicting for more than 5 billion annual peak sales and maybe more, using it as an IO backbone for the different combinations with AstraZeneca's oncology pipeline. So this could be definitely a major driver and also a source of financial stability that will allow us to continue and progress our programs. And I didn't discuss GSZ321 again. So I think this is what's nice about Compugen these days. We have financial stability that allows us to reach milestones from both internal and partnered programs. And each of those could be an amazing opportunity for Compugen. So we have definitely a few shots on goal here.
Terrific. Yeah. When I was first introduced to Compugen in 2017, it seemed that using artificial intelligence, machine learning, big data, and computational biology was fairly novel, at least it seemed to me. And now it seems much more commonplace. So, what competitive advantage does Unigen provide Compugen over the competition?
So there are a few aspects to that. I think one, people use AI and computational today. First of all, they claim many things, and not many have proven they can actually bring value, but definitely it's coming more and more. I'm not saying it's not. They're doing it along different phases of the R&D of a drug. It could be in the target discovery, in the drug discovery. There are many companies using AI for maybe small molecules at scale. And then also for clinical trial design, adaptive trial designs, analysis of clinical data. So people use it quite broadly, but there are not many companies that really go into the very beginning of this, which is the target discovery, to identify new targets that others are not following. Typically, pharma companies are tending, also biotechs, are tending to herd around the same targets. I don't know how many PD-1s. I'm sure we're going to see more and more RAS inhibitors. I mean, definitely. And we are trying to bring other targets. And we are not only trying. We have shown that we can identify such targets, and we are a validated platform, validated by our ability to bring such targets, validated by the collaboration we have around these targets. So I think this is one. The stage of the development that we focus on, which is the target discovery. But then it's also about being non-naive in how you use computational output. It's not only to use the platform and to know to use AI tools. This many people can do today. But really to have in one company the convergence of disciplines, to know how to be non-naive in the way you treat computational output, how to screen for such assets, how to know which asset to prioritize, and also how do you know which asset not to pursue and not to put too much efforts on. So I think also the know-how was built along years from computational discovery to the whole development built around such innovative assets is definitely another competitive advantage of Compugen that we're pioneer in this approach.
Okay. So, in your presentation, you simply talk about your pipeline, but it's undisclosed. So I assume it was developed using your Unigen platform, but how do you decide what to target with that platform?
So we always start with the end in mind. We always want to see, okay, which patient population we want to target, which mechanism as a whole we want to target that we think could make a difference to that patient population. And once we have a concrete answer to what are we targeting in the clinic, in the patients, then we start asking ourselves, what do we need to answer the question? What do we see in our database that would build along years? And based on that, we identify the actual drug target that we believe that it will target this one with an antibody typically, we could make a difference to that patient population.
I see. Are you able to share any information about how many targets in your pipeline or any that are getting close to preclinical development? What are your most advanced pipeline targets currently?
So in terms of numbers, we can say that, of course, we're talking about the funnel here, and we start
Yes.
with computational output. From the computational output, we already have quite a few targets in the pipeline, and then we have this funnel, as I mentioned before, one of our know-hows, how to funnel and screen, and then having obviously not too many candidates moving into lead selection, all of that. So the number it depends from where you count it, but it could be quite a few. In terms of how many are in advanced stage and giving specific guidelines on the early pipeline, so we typically avoid doing so on early assets. We prefer first to, before we commit to investor, to really unsolve the biology, de-risk the targets completely, and only then we disclose. For now, we don't have any specific guidelines on the stages in which we are, but this is the largest group in Compugen, and as mentioned before, we use our financial stability to continue to invest and leverage the Unigen platform to bring more such assets. This is the most I can say at this point.
Okay. That's fair enough. Well, let's talk about your COM701, and that's your potential first-in-class anti-PVRIG. What are the benefits of PVRIG, and why this particular trial design?
So maybe a few words about PVRIG. We identified it in a similar query to TIGIT, but when we started studying it, we identified that the biology is not only very different from TIGIT, it's very different from PD-1s or maybe any other checkpoint. So we talk about the target with a very unique biology, and due to that unique biology, we have seen signals with COM701 in places that typically checkpoints are less responsive. One of these indications, in which also the pathway is very dominant, and that's why it was a target indication for us to begin with, is ovarian cancer. And we treated patients in the last line, platinum-resistant ovarian cancer patients. And what we have seen in this difficult patient population, exactly, thank you for this slide. So if you look really to the end right, we had patients, some of them with 10 prior lines that failed everything. We see quite convincingly, even though it was not many patients, definitely not randomized study, that we could derive clinical benefit. It was in monotherapy and in combination with PD-1 or PD-1 plus TIGIT, that we could derive clinical benefit in these patients. When we did derive benefit, it was very durable and very safe. And therefore, we decided to monetize on these properties of COM701 and to take it earlier into the second, third line, as you can see in this slide. These are patients which are still platinum sensitive. They receive platinum. Sensitive means they're still responsive to the platinum. So they are much earlier than the patients we treated before. They have less compromised immune system. They have low tumor burden because we take to the MAIA study only the patient to respond. And our goal is to utilize the durability and safety we saw in the last line patients to prolong the progression-free survival of these patients, namely these patients that receive BEV and/or PARP, who have no standard of care. There is no option. They receive the 6 cycles of platinum. They cannot tolerate more than that. And basically, they have nothing to give them to prevent them from becoming eventually platinum resistant. There is no standard of care for maintenance. And this is the goal of this study, to show that COM701 can prolong the response to the platinum in patients who respond to the platinum, and by that, prolong eventually their life. So this is the MAIA study. It's a randomized study, blinded, in which we randomized 40 patients to receive COM701 in monotherapy. Definitely, we have a goal here to show clearly monotherapy activity of COM701, and we compare it to placebo, 20 patients. This is the MAIA study, and again, the readout is by Q1 27.
As I understand it, currently, because there is nothing for these patients, the median progression-free survival is under 6 months. Is that right?
The range of the older studies was 3.8 to 5.8. Actually, in the recent ASCO, we have seen the TIDOVA study. That is not the exact patient population we are studying it, but over there the PFS was 2.8 months. So the range could definitely be probably even lower than 5 months, but we are going to look into that depending on our patient population. But overall, yes, few months, and these patients are typically progressing.
So, what does success look like for this trial? What are you hoping to achieve in Q1 2027 when the top line data is released?
So eventually it will depend on the way the actual data will look like. It's a randomized study. So if the PFS of the placebo will be 3 months or 4 months, and this will also detect what success is. Overall, the goal will be looking at the curves and clearly seeing separation, suggesting that COM701 has monotherapy activity. It's not a pivotal study. It's not powered for significance, but definitely with this number of patients, we should have clear signal of activity.
Okay, all right. But you're not looking specifically for an additional 2-3 months, or is that something you're hoping for?
I think that if it will be 3 months, it definitely will be a success, especially, I mean, even if the PFS is 5.5 of the placebo, but definitely if it's shorter. If we're able to
Yes,
double from 3 to 6 months, it's going to be very meaningful. Less than 3 months could also be a success, again, depending how long will be the PFS of the placebo.
sure. So this is an adaptive trial. So assuming that you do have success in monotherapy, I understand that you'll then perhaps begin additional trials in combination with other immunotherapies. Is that correct, my understanding?
Yes, and I will add. First of all, our goal will be, after seeing clear monotherapy signal in this patient population, to start discuss with the agencies how we can get this drug approved for the unmet need of these
Yeah,
patients. Potentially, it could also be in monotherapy. So to think about registration trial. In parallel, after convincing monotherapy signal, since it's an adaptive trial design, we can easily add more arms. We can add an arm with BEV. We can add an arm with ADC. There are quite few companies struggling also with ADCs in ovarian cancer, and I'm sure they would like differentiation with a drug with a different MOA, which shows validated monotherapy signal. So I think that in parallel to moving into registration and discussion with the agencies on that, opening more arms could also open more opportunities indeed, and we can do it easily with the adaptive trial design we have here.
terrific, terrific. So assuming success, whether in monotherapy and or in combination, how big is the market potential?
So specifically for this population of the second, third line patient to receive BEV and PARP, we estimate eventually around 10,000 patients yearly, but definitely in this patient population, even if we just add BEV, it's already taking all the second line, so it means larger patient population. After showing results in maintenance in second, third line, why not thinking about first line? And we can also go later to protein resistant back in combination with Pembro and docetaxel that was recently approved. So I think that eventually, while the initial patient population is rather focused for the immediate potential registration trial, the option could be much broader, also outside of ovarian. We had signals in endometrial, breasts, colorectal, but we're focused now to show monotherapy signal, and then I think there will be many options for COM701.
And when you say 10,000 patients, where is that? Is that the United States, worldwide?
Worldwide.
What are your, so
It's around
worldwide?
an estimation, but yes.
Okay, terrific. So let's go next to Rilvegostomig. This is your partnership with AstraZeneca, and they're using Compugen's COM902 anti-TIGIT to develop their bispecific. And I understand this was pursuant to a deal signed back in, I believe, March of 2018 with AstraZeneca's development arm, MedImmune. So what's so interesting here is that until a few years ago, there have been a number of high-profile phase 3 failures in the TIGIT class from Roche, Merck, Gilead Arcus, and GSK Iteos, who mostly use an FC-active design of TIGIT. And now AstraZeneca's Compugen's TIGIT design, your COM902, with an FC reduced platform, seems to be one of the only TIGITs remaining in clinical development and may turn out to be a major success. So now we see AstraZeneca aggressively advancing Rilvegostomig, and I'm going to get a slide out in a moment. why do you think the COM902 will be successful where other TIGITs have failed?
So actually, we take that question and put it more on Rilvegostomig, which is the focus now. For COM902, we believe that there is some potential, definitely, and if Rilvegostomig is positive, it could also reflect on COM902. But Rilvegostomig, in specifically AstraZeneca's clinical program, has three attributes that are more specific for Rilvegostomig. So let's talk about Rilvegostomig, which is a bispecific antibody, first of all. So it's not only that reduced, which we believe is the right format for safety, for sure. I think it's proven to allow combination, and we'll go back to the combination in a second. But it's also the fact that they have shown that the bispecific has the potential, at least in a relatively solid preclinical settings with patient-derived samples, that they shown that rilvegostomig, the bispecific, is more active than just combination of PD-1 plus TIGIT. And we'll not go through all the biology around it, but we see around us some other bispecifics with cooperative binding that potentially are driving more activity than just combination. And it could be the case, and as seen here, also for rilvegostomig. So one is the antibody format, the bispecific, not tested before. Two is the clinical trial design. I think that AstraZeneca, and I'm not talking on their behalf, but from what I see on their program, that they have learned from the mistakes of others. They are powering the studies right. They are selecting for PD-L1 where it should be selected, like in non-small cell lung cancer, where some of them, like the recent study of Arcus that failed, that at least initially started as an all-comer study. They are looking at homogeneous population, and this is another aspect, the trial design, the number of patients, all of that. And thirdly is the combination strategy. They are doing combination that were not tested by others, mostly antibody drug conjugates with blockbuster drugs like Enhertu, like TROP2 ADC, like Dato, emerging data with other assets like the B7H4 ADCs, etc. So I think that their combination strategy really leverage the fact it's a bispecific antibody, which have less of a burden of contribution of components, and they can easily combine it with different assets and eventually get a win in a trial, in phase 3 study, potentially based on the combination, and not necessarily always just on comparing PD-1 plus TIGIT to PD-1. So I think these few attributes are making this program very different from the others who failed.
I see. I put up the slide that you had in your corporate presentation, and it shows here, I guess, on the right-hand side, some of that anti-tumor activity. And as you know, sort of confirming what you just said, that it looks like the bispecific anti-tumor activity is greater than the sum of its parts.
Exactly.
Okay. So just going back a couple slides, if I may, this is the slide summarizing all of the, I guess these are 25 trials that Rilvegostomig is being used in by AstraZeneca, which is quite a few. And I see most of the phase 3 trials are expect to read out, I guess, after 2027. Is that your...
Yes. So first, difficult to see, but this slide definitely reflects the magnitude of effort that AstraZeneca are putting into this drug in combination with different assets, 11 phase 3 trials as mentioned. And this is even without the other trials. For example, now in ASCO, there was an investigator-sponsored trial, the I-SPY 2.2, which had a late-breaking abstract, also showing very promising activity for rilvegostomig plus Enhertu in early line breast cancer, which is not even here yet in this trial. So I think the development is even broader than that and have the potential to go broader than that. And about the phase 3 readouts, so by AstraZeneca, it's post 27. They didn't give guideline if it's 28 or 29. I think it's very probable, from my understanding, is that some of the trials could read in 28. They could also read in 27, interim analysis, etc.
Okay, sure.
But per AstraZeneca guidelines, it's 28 or later, meaning after 27.
So approximately how many patients are enrolled by AstraZeneca in these various rilvegostomig trials?
I don't have the exact numbers, but these studies, especially the phase 3, many of them are 800 patient studies. Some of them are more. So we're talking about thousands and thousands of patients.
I see. Do you have any ballpark idea of what AstraZeneca is investing in these trials?
I don't have an exact or even a very good approximation, but we know that these kind of oncology trials, the cost per patient is we're talking about tens of more probably of thousands. So we're talking about huge investment from AstraZeneca side.
Okay, fair enough. So, I mean, this is potentially blockbuster numbers that AstraZeneca has been projecting in its own presentations for Rilvegostomig. And that could translate into a pretty big number for Compugen. Yet it doesn't seem to be reflected in Compugen's market cap. Do you have any explanation?
So I think that people are just after seeing so many failures of TIGIT, probably are not even bothering to go too much into the details of the points I mentioned before, the bispecific format, that part of some of these studies design are not even comparing directly Rilve versus Pembro. I think that eventually people are skeptical and they want to see a phase 3 success. Some of them with the accumulation of more, and we just had now again in ASCO, a nice results in another study, but non-randomized, and still over survival, which compares very favorably to historical control that reflects on an ongoing phase 3. So I think some with the accumulation of phase 1-2 data will start to be more convinced. AstraZeneca in their ASCO event just mentioned the fact that they do see and refer to that specific study in the hepatobiliary tract cancer, that they see this durability of responses using Rilvegostomig. So some will be convinced with the accumulation of early data, and some will wait to the phase 3 success.
Okay. I think that's probably fair enough. You know, what I found interesting was back in December, just a few months ago, I guess that's now 6 months ago, Compugen announced a, um, a deal where AstraZeneca paid $65 million up front and a potential additional $25 million to buy back what appears to be a very small portion of the future royalties payable on the commercial sales of Rilvegostomig. And just want to get your comments on it, because it seems that your agreement before this deal was announced that AstraZeneca was going to pay mid-single digit royalties to Compugen for its COM902 as part of the Rilvegostomig bispecific. And after the deal was announced, the royalty payments were still mid-single digit. So what was going on there, and how did this come about?
Yeah, so mid-single digit is not an exact number. It doesn't necessarily mean 5% as is. There is a small range with what is considered mid-single digit. So you read it right. It was mid-single digit before. It remained mid-single digit after. We cannot disclose the exact numbers because typically pharma companies don't allow us. But you're right saying that was a very small portion of our future royalties. On the one hand, this deal allows us now to have cash runway into 29 to reach all our internal and major partnered potential milestones. And in parallel, we maintain value, and we still have mid-single digit. And if this drug going to be the blockbuster we hope and think it will be, it's going to be a major, very important for Compugen. We didn't lose a lot value from Rilvegostomig.
So how did the deal come about? Like, who was approaching who, if you don't mind sharing? Because it seems to be a very good deal for Compugen. I'm not sure exactly what AstraZeneca got out of it.
AstraZeneca eventually got out of it. I mean, the fact it remained mid-single digit after doesn't mean that they didn't receive any part of the royalties. They
Sure,
received
of
a
course.
small part of the royalties. If they are estimating it to be an IO backbone for new combinations, a potential blockbuster, so I think definitely I can reassure you that AstraZeneca did this deal because they think it's a good deal for them. And for us, it was the right deal in the right time, as I mentioned before. So I think it was a very good deal for both sides because it was a win-win for both sides.
Okay, terrific. I mean, currently Compugen has a market cap of $200 million, and that takes into account about 135 million of cash as of March 31 and no debt. So yeah, just an interesting development on valuation of the Rilvegostomig asset. Let's now talk about your deal with Gilead for GS0321, which was formerly COM503. This was a deal that was signed with Gilead in December of 2023. And let me just get to a slide on this one. This looked like an attractive transaction, but I just put up the slide for what GS0321 is, and perhaps you can talk about it. This was a preclinical asset, and it looked like you got, well, 60 million up front and 30 million upon IND clearing. For a preclinical asset, this looked like a very lucrative deal, and of course, milestone payments of another $750 million. So can you talk about why this preclinical drug looked so enticing? And how the deal came about?
So I think this is another demonstration for how our computational engine, Unigen, can identify new biologies that no one have shown anything similar to that even by concept before. In this case, we identified computationally that in the tumor microenvironment you have this cytokine called IL-18. It's a soluble factor that can activate the immune system very effectively against cancer, but it's floating there inactive because of another soluble factor called IL-18 binding protein that binds it in high affinity and prevents its activation. So what we identified that in a tumor microenvironment, there are high levels of this complex floating there in an inactive form, and it's very high in the tumor, very low in other places. So in this case, we developed a differentiated approach. Instead of using a cytokine, like people are trying for ages to use cytokines and to engineer them to treat cancer, and it's approved but very toxic and not really used at scale, we identified a new approach. We developed an antibody that inhibits the inhibitor, release IL-18 to perform its activity in the tumor, and by that, and the reason that Gilead and actually some other pharma companies were chasing this molecule is in such a nice deal on the preclinical assets, because preclinically, at least we have shown that we have the potential activity of a cytokine without the side effects of the cytokine, at least with minimal side effects preclinically. And this is promising. This is novel mode of action that could really make difference for patients. And the clinical trial is ongoing, and we're very excited to see how it will develop.
Okay. So as I recall, under the agreement that you entered into with Gilead, it's Compugen that is paying for and running the phase 1 trial. Is that right? So I took a look at clinicaltrials.gov this morning just to see what was going on with that trial. So it looks like the phase 1 trial, you're looking at safety and tolerability in monotherapy and in combination with the PD-1 that's owned by Gilead in various advanced solid tumors, and you're looking at identifying the maximum tolerated dose, the administered dose, and the recommended phase 2 dose as monotherapy and in combination. So where does the trial stand right now? Are we, and I see it looks like an estimated 200 patients, so it's not a small trial.
Yeah, so, the numbers in clinicaltrial.gov don't always reflect exactly, but
Sure.
overall, it's a typical phase 1 of monotherapy with some backfill cohorts and combination, and then expansion so this is the second stage of the phase 1, expansion into a few different indications in mono and combinations. The trial started at the beginning of last year. It's moving really nicely, and we didn't give guidance on the exact place where it stands. Yeah, but it's again a typical phase 1.
Are you happy with the pace of enrollment?
Yeah, it's moving very nicely.
Okay. And in this trial in particular, because of your relationship with Gilead, I take it they control the release of the data. And one of the things that I'm always mindful of when I see these agreements with smaller biotech companies like Compugen is it's so important to Compugen to get data out there and probably less significant to Gilead. So how do you deal with that tension? Like how much control or influence do you have in being able to release data on an interim or whatever basis?
Yeah, so first of all, yes, you are right that when we disclose data, we do have to discuss with Gilead, and this should be in collaboration, in a common decision. Gilead are a great partner. We work very nicely together. They are aware of the fact that we need to disclose data. They also disclose them safe. They recently disclosed, for example, their CCR8 phase 1 data. So it's not that they are not disclosing early trial data. And eventually it will be discussion. But they understand that we are biotech and we need to release data, and they are good partners.
Okay, that's a terrific answer. So, just from a corporate summary point of view, I just remind listeners that Compugen trades both on NASDAQ and the Tel Aviv Stock Exchange, and the symbol is CGEN. And when I was looking at your filings, it looks like the shares outstanding, at least as of March 31, 2026, was just 94,553,000. You've got a market cap as of the close of Friday of about $200 million. Share price has been trading just over $2, and I guess your 52-week share price range has been from a low of $1.30 up to a high of $3.24. So if we can summarize the upcoming catalysts in the next 12 to 24 months, what do you see those as being?
So we have the COM701 MAIA results, in which we will have the PFS data, which is the key data we're looking for, by Q127.
Okay.
We have more data from AstraZeneca. Again, we just seen in ASCO, and I'm sure they will release more and more data moving forward. COM503, not disclosed yet. We didn't give guidelines, but definitely there could be release of COM503 data. Same goes for the early pipeline. So this summarizes the pillars of our current R&D pipeline.
That's terrific. So one of the challenges in the biotech space, and especially with pre-commercial companies, is biotech's been beaten up pretty badly in the last number of years. A bit of more of a recovery more recently. But how do you make sure that shareholder value is protected, and especially where there can be sort of large gaps in data release?
So we do put efforts to maintain data release as much as we can. Eventually, success will also bring release of good data. So we work heavily to bring more promising preclinical assets far enough and then disclose them. We work heavily on execution across our studies. AstraZeneca gladly are also working very heavily on Rilvegostomig. So eventually, I think there will be accumulation of data. In general, I think these are very exciting times for Compugen. We are being an actual clinical stage company. We have a very strong foundation of computational platform that was built along years, and you described it nicely at the beginning. But we're actually a clinical stage company that uses its AI platform to identify and develop our drug targets only around a decade, a bit more. So after around a decade of being a clinical stage company, we have hopefully having the foreseen future and approval of our first asset that we discovered ourselves computationally years ago, Rilvegostomig. We are very close for a proof of concept for another molecule, which is a first-in-class asset, the COM701. And again, we have the cash and the financial stability to continue leverage our pipeline and build more such assets. So I think these are very exciting days for Compugen, and the data will continue to accumulate a long time.
Terrific. We've covered quite a bit this morning. Any other topics that you'd like to cover?
No, thank you. I think we covered it really nicely.
Oh, terrific. So for full disclosure, I am independent of Compugen and receive no compensation from them for making this podcast. I do own shares in Compugen, which were purchased more than a year ago. This podcast is for information purposes only and should not be considered a recommendation to buy or sell Compugen shares. So, Eran, thank you so much. It's really been a pleasure.
Thank you. Pleasure as well.